place where all the solids, remember, everything that can be digested is now digested. Now all the solids is passed to the colon, which moves like this. If you take your right hand and you move in a circle like this, you are moving with your colon. Okay. This part is called the cecum, and there you see the appendix. And the colon is a long time for the solids to pass. And the reason is the absorption of water is taking place here. So it tries to get all the water that is left in this undigested food to use it for your body and not release it with the feces. Now, okay, undigested solids or fibers and the water is absorbed. Also vitamin K and B are reabsorbed with the water. It is also taken up. And then the solid waste will be stored in the rectum, and then it will be defecated or egested through the anus. I want to ask you, if you have diarrhea, what is the main component of your feces if you have diarrhea? Water. Do you want to lose that water? So that is why diarrhea is dangerous now, because it makes your large intestine, your colon, not able to absorb water. Interesting, I want to tell you why. The bacteria that cause diarrhea makes the inside of your large intestine giving off a lot of sodium ions. When there's lots of sodium ions, it changes the osmosis, and all the water that can be absorbed into your colon cells will move into the space where there's lots of ions. You see? And now you secrete lots of water. Your absorption of your vitamin K and B is not well. It's also egested or defecated with the feces. And you dehydrate. You have pain because sometimes it causes pain. And that is why diarrhea is not a good thing to have. How do we treat it? You make sure that you take in enough fluids. As many fluids as you can to keep hydrated. And then, of course, it is a bacterium many times. So what will you drink? Antibiotics. They give you antibiotics for gastroenteritis when you have inflammation of the large intestine, which causes diarrhea. Okay. Right. So there we see the function of the large intestine, reabsorb water. If you do not absorb enough water, it leads to diarrhea. If you absorb too much water, then the feces is very hard and you become constipated. So that is why we take in what? What do we take in to prevent constipation? Fiber, which is what part of the plant? The cell walls, cellulose. Because the cellulose keep enough water to keep the feces soft. That prevent constipation, prevent cancer of the colon. And with the peristalsis, it can clean out all the solid waste from your body. Okay, right. And I already told you about the bacteria, but not all bacteria are bad. We have Escherichia coli, and they need to be in your body. They need to be in your body. E. coli. because they help your body to produce your vitamin K and your vitamin B. And what is so interesting is the Escherichia coli, those friendly bacteria, they can cause the diarrhea. Ne? If there are too many, they try to be the bosses in there. And then they can also cause diarrhea, E. coli. Okay. But they are helpful and they need to help to be there. But, ne? They also generate the gases. Okay? The bacteria in the large intestine create those gases. Ne? Okay, right. And then the rectum. There we see the rectum that needs to eliminate the undigested material. We know it is the cellulose of the plants which produce the roughage and the fiber. Lots of bacteria in that part. I also want to show you the appendix, which is there a little piece. And when you have appendicitis, I-T-I-S at the end of a word, I-T-I-S refers to an inflammation, where you have bacteria or a pathogen taking over a part of your body. Appendicitis. Now, is where there's too many bacteria that gather there and it causes lots of gases, pain, and they remove that part. It is a vestigial part of the body, which they say long ago, before people made fire and cook and have preserved food, they eat a lot of rough fibers. You know, when you cook your meat, it's already easier to digest. And there must have been some help in the digestive processes. Okay. There's the word vestigial organ, which is an organ that does not have a function now, but research shows that it, like our wisdom teeth, is also a vestigial organ. It is now drawn out and we do not use them. But it was necessary when we had to eat our food.
This broadcast is made possible by the Capricorn Group.
ShopRite has always been here for you, and our promise to bring you low prices on what you need most will never change. Get 2 kilos Nam milk or Mayere assorted, only $34.99 each, and 175 grams Dettol bath soap, just $10.99, only at ShopRite. One stop, one safer shop.
Biology. Yesterday we did the nutrient carbohydrates. Any questions that you need to ask today on carbohydrates? I want us to quickly revise. Carbohydrates will give how many energy per gram? How many energy per gram? 17 kilojoule per gram. Why do we eat? Why do we eat carbohydrates? For energy. That is the main reason why we take in carbohydrates. The elements for carbohydrates? C, H, and O. The ratio of hydrogen to oxygen. The ratio, hydrogen to oxygen is 2:1. We can look at this molecule and we can count the C, the H, and the O. Now, I have 3 types of carbohydrates. 3 types. Monosaccharides and disaccharides and polysaccharides. What is the simplest form of carbohydrate that I can have in my hand? And it's still a carbohydrate. A monosaccharide. Nee. So it means that I have 1 molecule. 3 examples that you know of monosaccharides: glucose, fructose, and galactose. Good. If I want a disaccharide, I need to connect 2 monosaccharides. The reaction to form a disaccharide, condensation. Well done. I told you, you are clever. We just have to sort it in the correct files and know when to take it out. Are you with me? So, when condensation takes place, I need a specific name of a bond. We call that a glycosidic bond. The byproduct is water. Good. Okay, and there we have it. Thank you, class. Now, when I want to break it down again, if I want to move from maltose back to glucose plus glucose, what do I need to do? I need to split a water, do you agree? I need to take a hydrogen from water, reverse it into that spot, and the OH, put it in that spot. So that means I need to split water. What do we call that reaction that splits water? Hydrolysis. The term Lysis, lysis, always refer to split water. So, when I have a maltose molecule and I need to split it, I need to split the water, and therefore, from maltose back to glucose is hydrolysis. Good. And now I have to split the water. Water was the byproduct when condensation takes place. To reverse it, I need to split the water and add it back on the correct spots. Good. Okay, let's continue to the next, and I think we do understand that if I add the monomer to, ek gaan nie daai ene gaan, daai het ons nou klaar gedoen, sorry, I want to continue to that one. If I am looking at the different types of carbohydrates, and a monosaccharide plus a monosaccharide will give me a disaccharide. Glucose plus glucose gives me maltose. Lots of glucose in a long string give me a polysaccharide. It can be starch. We are revising. Starch I will find only in plants or animals. Plants. And that same storage molecule of lots of glucose in an animal is called glycogen. Where do animals store the glycogen? Muscles and liver. Muscles and liver. Now, why do I have a small molecule and then I have a large molecule? I want you to think. if I take glucose and I put it in water, it will easily dissolve. It quickly happens. You agree? But if I take starch, it's a very large molecule. It will not dissolve in water. Monosaccharides and disaccharides quickly dissolve in water. Nee. But a polysaccharide, starch, will not dissolve in water. So, in which one will I store something? In something that quickly dissolves, or in something that cannot dissolve? Something that cannot dissolve. Okay, so I use the polysaccharide as a storage molecule. Why? It is insoluble in water, and therefore it has no osmotic effect. You remember that picture that we always draw. And I'm taking you back to transport through or between cells. Ne? I want you to think. I have two cells. They are very close to one another. And inside this cell, I have a high water potential. I want to make this cell having a lower water potential. What will happen in this cell? What will I add to this cell if I want this cell to have a lower water potential? Salt or sugar? Yes. But will I use a polysaccharide or a monosaccharide? Yeah, I want to the water potential. Will I put in something that can dissolve or something that cannot dissolve? So I will put in a monosaccharide. Now, so let's add some sugar in here, right? I add some sugar in here, and this means that this will lower the water potential. In which direction? A to B or B to A, I will have osmosis. B to A. Do you follow me? So the moment when I have glucose and maltose in a cell, it will have an osmotic effect. But if I have a polysaccharide, it will not dissolve, it will have no osmotic effect, and therefore it's good storage material. Okay, it is a large molecule. It will take a long time to digest. That is something I want you to remember. That is the reason why polysaccharides are good for storage of energy. So if something digests easily, I will not store something in something that's breaking down. I will store something in a large molecule that will not break down, that will not cause osmosis, that will not dissolve in water. That is a good storage material. Okay, then cellulose. Cellulose is made out of, or is making up cell walls. Do we get energy from cellulose? No, we do not. But we do use the cellulose as the fiber in our diet. Nee. So cellulose is definitely also a carbohydrate. Yes, then we did the starch test. Okay, and we know the starch test was iodine, drops of iodine solution. It is orange, and if starch is present, it will become blue-black. You remember? Good. And then the Benedict's test, blue. If there's a small amount of reducing sugar, green. If there's a large amount of reducing sugar, it will turn to orange. Okay, Right. Are you good on this? Let's quickly continue to proteins. So now, let's think protein again. So, if we turn in our books, let's see now proteins. First, we want to find out what are the elements that we see in a protein. What are the elements? Oxygen, carbon, hydrogen. If we have oxygen, carbon, and hydrogen together, we know it's organic. You remember? But we also have nitrogen for proteins. When I look at a protein molecule and I want to see the smallest piece of molecule that I can use to build a protein, I have an amino acid. The amino acid is the smallest unit that I can use to build a protein. If I have one amino acid and I add another amino acid, I have a dipeptide. Ne? One amino acid, two amino acids, dipeptide. Three amino acids, tripeptide. Okay? Do you hear the word protein? Not yet. Now, if I have a long string of tripeptides together, I have a polypeptide. Do you hear protein? Not yet. We did something about that when we did enzymes. A protein is only a protein when I have a specific string of amino acids folded into a specific shape, tertiary structure, then making a three-dimensional structure, then I only have a protein. So, the building blocks of proteins we will call amino acids. Right. Are you with me? We have the elements CHO and N, and then we can have S for sulfur, P for phosphorus, Fe for iron, which can, in different proteins, be part of the molecule. Are we good? Yes? Okay. If we look at the amino acid, we see on the one end an amino group, NH3. On the other part, we see a carboxyl group, COOH actually. Now, but there we see the carboxyl group. That is important to understand. We do understand the elements, and now, if I want to link two amino acids, I need to go to the amino group and to the carboxyl group. HO will form water, you see, and there I can see condensation takes place. What do I want you to remember? Higher level, you need to know, for two amino acids to join, the carboxyl group and the amino group needs to form a bond. The bond is a peptide bond. It is a condensation reaction and it forms water. Okay. And if I have two amino acids, I have a dipeptide. And if I have three amino acids, I have a tripeptide. If all the amino acids link together in a long chain, I have a polypeptide. Good. Right. So, from 4 to 50 amino acids can be a polypeptide. But it is not a protein. Nah. A polypeptide is not a protein. Okay? Okay. So, in order to form a large molecule, we know that carbohydrates can be a macromolecule, but proteins is also a macromolecule, which means monomers need to join together to form a polymer. The monomer of carbohydrates is monosaccharides. What is the monomer of a protein? The one that I can link to build a protein? Amino acid. The amino acid will form the smallest building block. If I link them together, I can end up by building a protein. And here we are. Proteins has a primary structure, which means I have a specific sequence of amino acids. If they are then with hydrogen bonds, either link as a sheet or a helix, I have my secondary structure. My secondary structure needs to be folded in a three-dimensional structure. Then I have the tertiary structure, and at this stage, I can call it a protein. Some protein need a quaternary structure like hemoglobin, where the group need to add the iron groups to make it the complete protein.